Hey there, science fans. Today we're diving into the world of ELISA, a powerful tool that helps us diagnose infections. ELISA stands for Enzyme-Linked Immunosorbent Assay. It's a mouthful, I know, but don't worry, it's not as complicated as it sounds. Think of ELISA as a tiny detective in the lab. It helps us uncover clues about infections by detecting antibodies in our blood. Antibodies are like our body's defense system. When a virus or bacteria enters our body, our immune system produces antibodies to fight them off. ELISA uses a special plate coated with antigens. Antigens are like wanted posters for our immune system. They help our body recognize and target specific invaders. When a blood sample is added to the ELISA plate, any antibodies present will bind to the antigens. This binding triggers a series of reactions that produce a color change. The intensity of the color change tells us how many antibodies are present. This helps doctors determine if someone has been infected with a particular virus or bacteria. Pretty neat, huh? Now, ELISA isn't just one test, it's a whole family of tests. Each test is designed to detect specific antibodies or proteins associated with different infections. Let's look at a few examples. First up, we have IgM ELISA. IgM antibodies are the first responders of our immune system. They appear early on during an infection, so if you test positive for IgM antibodies, it usually means you have a recent or current infection. Next, we have NS1 ELISA. This test is specifically used to diagnose dengue fever. NS1 is a protein produced by the dengue virus. Detecting NS1 in the blood is a strong indicator of a recent dengue infection. Finally, we have NAT, which stands for Nucleic Acid Amplification Test. This test doesn't directly detect antibodies. Instead, it looks for the genetic material, DNA or RNA, of the virus itself. NAATs are highly accurate and can detect even very low levels of the virus. Let's take a closer look at how ELISA helps diagnose dengue fever. Dengue diagnosis is often a tale of two phases, the acute phase and the convalescent phase. The acute phase is the early stage of the infection, usually within the first week of symptoms. During this phase, doctors may use IgM ELISA, NS1 ELISA, or NAT to confirm the diagnosis. If the acute phase tests are negative but dengue is still suspected, doctors may request a second blood sample during the convalescent phase. This phase occurs about 7 to 10 days after the onset of symptoms. During the convalescent phase, IgM antibodies are usually detectable, so a positive IgM ELISA result at this stage can help confirm a recent dengue infection. While ELISA is a valuable tool, it's not foolproof. One challenge is cross-reactivity. This happens when antibodies produced against one virus also react with another virus. Think of it like this. Imagine two people wearing similar hats. Our immune system might mistake one person for the other based on the hat alone. This can lead to false positive results. Cross-reactivity is a common problem with flaviviruses, a family of viruses that includes dengue, Zika, and yellow fever. These viruses share some similarities which can confuse our immune system. To overcome this challenge, doctors often use a combination of tests and consider the patient's symptoms, travel history, and other factors before making a diagnosis. Analyzing ELISA data can be tricky, especially when dealing with complex infections like tuberculosis. But fear not, science fans, a new method called ELISA-R is here to save the day. ELISA-R is like a superpowered calculator for ELISA data. It uses advanced mathematical models to analyze the results more accurately. This helps reduce errors and improves the reliability of diagnoses. Traditional ELISA analysis often relies on a single measurement called the endpoint titer. But ELISA-R takes into account multiple factors like the shape of the curve and the slope of the line to provide a more comprehensive analysis. This new method is like upgrading from a magnifying glass to a high-powered microscope. It allows us to see the finer details and make more informed decisions. Tuberculosis, or TB, is a serious infectious disease that affects millions of people worldwide. ELISA has been a valuable tool in the fight against TB for many years. One of the main challenges with TB diagnosis is that the bacteria can remain dormant in the body for years before causing any symptoms. This makes it difficult to detect the infection early on. ELISA helps overcome this challenge by detecting antibodies against specific TB proteins. These antibodies can be present even when the bacteria are dormant. ELISA is also used to monitor the effectiveness of TB treatment. By tracking antibody levels over time, doctors can see if the treatment is working and adjust it if necessary. The recent COVID-19 pandemic caused by the SARS-CoV-2 virus highlighted the importance of rapid and accurate diagnostic testing. ELISA quickly emerged as a valuable tool in the fight against this novel virus. Scientists and researchers around the world worked tirelessly to develop and validate ELISA tests for SARS-CoV-2. These tests detect antibodies against different viral proteins, such as the spike protein and the nucleocapsid protein. ELISA tests for SARS-CoV-2 have been used for various purposes, including diagnosing current infections. ELISA can help identify individuals with an active infection, even if they have mild or no symptoms. ELISA can determine if someone has been previously infected with SARS-CoV-2, even if they never developed symptoms. ELISA can assess the immune response generated by COVID-19 vaccines and track antibody levels over time. You might be wondering, if ELISA can detect antibodies, why not just use it to test for IgG antibodies all the time? IgG antibodies are the long-term protectors of our immune system. They stick around long after an infection has resolved. While IgG testing is useful for determining past infections, it's not ideal for diagnosing acute or current infections. This is because IgG antibodies take time to develop. During the early stages of an infection, IgG levels may be too low to be detected by ELISA. This could lead to a false negative result, meaning the test says you're not infected when you actually are. That's why doctors often use a combination of IgM and IgG testing, along with other clinical information, to get a complete picture of the infection. So there you have it, science fans! ELISA is a powerful tool that helps us unravel the mysteries of diagnostic testing. By detecting antibodies and other markers of infection, ELISA allows us to diagnose infections. ELISA helps identify the cause of an illness, even when symptoms are vague or overlapping. ELISA can be used to monitor the spread of infectious diseases in a population. ELISA is essential for evaluating the effectiveness of new vaccines and therapies. As scientists continue to refine ELISA techniques and develop new applications, this versatile tool will continue to play a vital role in protecting public health and combating infectious diseases worldwide. Keep those lab coats on and stay